Hello, everyone. Can you guys hear me?
Yes, we can hear you.
Yes, sir. Excellent. I'm ready when you are.
Good evening, one and all. Hope all of you are doing well. I would like to start today's evening with a quote of Tim Minchin, which goes as, "Science is simply the word we use to describe our method for organizing our curiosity." The Department of Chemistry, St. Xavier's College, Autonomous, Kolkata, welcomes you all to RACB 2022 International Chapter 1, a series of international webinars for answering your curious minds. An overwhelming response for the webinar from our participants is the evidence that the eagerness to know and explore had not come to rest in spite of the pandemic. Before commencing today's session, I would like to give a brief introduction about RACB. Recent advances in chemical and biological sciences, abbreviated as RACB, is a series of webinars based on the latest research and works in chemical and biological fields, which are being conducted by eminent scientists of the generation. It had been organized by the Department of Chemistry of St. Xavier's College, Kolkata, since 2019, and we are back again, but now in the virtual mode. So without any further delay, I would like to move on to today's session and call upon Dr. Ankur Ray, HOD, Department of Chemistry of St. Xavier's College, Kolkata, to address our enthusiastic audience.
Good evening, respected Father Principal, and good evening, everyone, and a very good morning to Professor Chakraborty. Welcome to Recent Advances in Chemical and Biological Sciences 2022, Chapter 1. RACB is a series of webinars on the latest trends that evolved in the field of chemical and biological sciences and how these two fields converge to explain the most exciting phenomena in nature. This event has been organized to enrich the young minds so that they can navigate their way through the path of science. With these words, I would like to call upon our respected Father Principal, Reverend Dr. Dominic Savio, to deliver the inaugural address of RACB 22, Chapter 1. Please, Father.
So thank you, Dr. Ankur Rai. A very good evening to all of you. Of course, a very good morning to Dr. Sourav Chakraborty. So once again, very warm welcome to all of you present here today for the inaugural ceremony of the 2022 edition of Recent Advances in Chemical and Biological Sciences, a series of webinars by eminent speakers around the world, organized by the Department of Chemistry, St. Xavier's College, Autonomous, Kolkata. In today's times, when there is an ardent effort to restore our lives and the world around us to normalcy, the pursuit for science cannot be left unfinished. With our inquisitive minds, we can quench the thirst for scientific knowledge through newer means amidst the much altered scenario. The participants of the event will get an insight into varied topics of interest in the frontiers of chemical and biological sciences through engaging sessions chaired by esteemed scientific minds across the globe. We have with us today Dr. Sourav Chakraborty, the Central Connecticut State University, USA, and today and tomorrow surely we have Dr. Develina Samantha of the University of Texas at Austin, USA, who is an alumna of our college. I want to thank the speakers for accepting our invitation in a special way today. I would like to thank Dr. Sourav Chakraborty for making himself available for us this evening, for him this morning, for the a very interactive session. I hope that event will be an overall success, and I take this opportunity to extend my best wishes to the organizers of the program, the students, headed by Dr. Ankur Ray, who with their untiring efforts have envisioned and brought this idea to fruition. With these few words, I declare RACB 2022 International Chapter 1 open. Wish you all the best. May God bless you. Thank you.
Thank you, Father.
Thank you, Father, for such an overwhelming inaugural speech. Without any further delay, I would like to call upon our esteemed professor of the chemistry department, Dr. Shonjeev Ganguly, to welcome our speaker for the day.
Good evening to all of you. Good evening. Sourav Chakraborty. There is some problem in hearing. Can you hear me?
Yeah, Shonjita, you're audible.
Yes, you're audible, sir.
Okay. There were some network issues, that's why. So it's my pleasure to introduce someone I know for quite some time, maybe for the past 20 years, I think. So it's none other than Professor Sourav Chakraborty, Assistant Professor of the Department of Chemistry and Biochemistry, Central Connecticut State University, USA. Well, he completed his graduation, BSc from Calcutta University, Narendipur, and MSc from Forest Research Institute. He then went on to complete his PhD from New Orleans with a thesis on molecular probes for biologically important molecules, which contain a study of thiourea, hydroxyl radical, peroxynitrates, etc. Throughout his career, he has done a variety of scientific work, and from studying the effects of water availability on emerald ash borer larval performances and phloem phenolics of Manchurian and black ash to mapping oxidations of etc. Well, many other things. So he has also worked on the impact of volatile and non-volatile constituents of coffee on its aroma quality. Most importantly, he has won the first place in UARDC annual poster competition at postdoctoral level in 2012. He is also currently a part of the American Chemical Society. Now with this brief introduction, may I now request Professor Sourav to deliver his speech.
Thank you, Shonjeev. A few minutes ago, Father said eminent. Whatever I am, I'm not eminent. I'm just a normal, regular guy who is interested in asking questions. And Shonjeev would know, because for 20 years or so, I have been asking questions which sometimes are likable, sometimes they are not, because it bothers people when you constantly ask questions. And those questions, some of them I don't have answer to, but I'm ready to figure it out. That's what science is to me, and that's what science would be for you. You would like answers for some of them you won't like answers, and then you will ask more questions. Let me share my screen first. Let's see, this is. Is the screen visible? Can you see it?
Yes, sir.
Yes,
Okay.
sir.
Thank you. Go ahead, get started. So first of all, I'd like to thank Father, Organizing Committee, Dr. Ray, and Dr. Ganguly for this nice introduction, giving me the platform to share my research with you. I have been in the field of chemistry and in quite a few other fields for last 15, 16 years. Most of my research is interdisciplinary. I am interested in solving biological problems using chemistry or the knowledge of chemistry we have. In this particular session, I will talk to you about polar metabolite detection. These are relatively challenging projects, even if it seems very simple at the beginning. And I will essentially give you some information about new technique that we are working on called hydrophilic interaction liquid chromatography. I'll give you the tenets, and I will also let you know what are the caveats in the process that we all should be careful about. And then we'll share some data related to the interdisciplinary research that we do here at Central Connecticut State University. But before we go forward, let me give you a bit introduction about my university. We are centrally located in New Britain. This is near Hartford, Connecticut. Our university was founded about 170 years ago. This was a teacher school and then changed its status about 40 years ago and turned into a university. It's a regional university of this area. Pre-pandemic, we have about 13,000 students. 11,000 of them were undergraduate and 2,000 of them were graduate students. We run about 51 graduate programs in our university, although chemistry and biochemistry is strictly undergraduate program here. We have reasonable faculty strength and diversity in research areas. My colleagues work on analytical, pharmaceutical, different type of biochemical, inorganic and organic chemistry related research. Now, just to give you an overview, I prepared a map. I thought you would enjoy looking at the university from a bird's eye view. So let me just play a quick video for you. This is our campus. If you take a look from the above. This is sprawled. It's essentially one part of the New Britain, and you're looking at variety of different building, athletic field, student dorms, and then eventually here is the building where we typically work. Now it's zoomed out to show you where we actually are, and as you can see, this is an exploratory map. We are right between, this is Long Island, which is very close to New York, and that's where Boston is. And we are right here in the middle. So about 2 hours from New York and 2 hours from Boston. It's very cold in here. During the winter time, the temperature goes to often single digit in Fahrenheit. So about minus 20 degrees Celsius, minus 23 degrees Celsius. And if we look at a two-dimensional map, this is where we are. Here is New York, here is Boston. so right in the middle. My lab's research team, as I mentioned before, is interdisciplinary. I am personally interested in analytical biochemistry. I do work in the area of food and plant biochemistry. I do method development, and I have some interest in biomaterials. This particular research is specifically interesting to me because I am generally interested in carbohydrate metabolism. And a few years back, there was a chance meeting. I was in the elevator. One of my colleagues from biology was moving some plants. And he was going to the top floor of this building where the greenhouse is. And I liked the plants that he was carrying, and I asked him what they are. And he said, this is a unusual species that he's working on for last about 20 years. And this species generally comes from southern part of United States as well as Latin American countries. And then seeing my interest, he asked me to come by and take a look at the plants. And I got fascinated with the nectar that he had. That nectar was colored. And he told me, let me show you a picture. He showed me this plant and the flower and the nectar, and he told me that nobody knows why this nectar shows pigmentation. So that was the start of this research. Now, for a very long time, I have been working with trees. And leaf carbohydrate metabolism is an important factor when we consider plant-insect interaction or plant-fungi interaction. I talk about leaf as if it's all broadleaf species. It's not. Conifers have very similar type of mechanism. So it's very broad in terms of how carbohydrates change due to abiotic and biotic stressors. So the two terms, constitutive and inducible defenses, are related to any attack that a tree get when it's in its natural environment. In both of these cases, what we were really looking for is the profile of simple sugars, things like glucose, sucrose, and fructose. In a few minutes, I will tell you more about nectars, and you will see why this particular nectar is kind of unique. Now, I want to introduce you to some people without whom I would not be able to give this talk. They came forward, they helped me throughout the process. At the early part of this project, Ashley and Endora took up this project, started working on the initial part, and then me did some developmental work related to this type of chromatography. And for past 1.5 years, Julia is carrying this work forward. She's currently a senior and will be graduating by the end of this year. And then we should be perhaps able to get this data out because I think we are very, very close. Two other people worked on this project. Unfortunately, I don't have a picture for them, but Amela and Mache both did work in between these two groups of people where they developed part of the method. Tom Mayon is my colleague in biology who grows these plants from cuttings here at Central Connecticut State University. He also travels to different countries down in South America where he gets the seeds or other materials to kind of explore what the species distribution is, what the biology is, and how this particular species is different than other species that grows in that region. Carl Griffin is a chemist who works at Agilent Technology, and hearing the type of research I have been doing, at the beginning of the project came forward and helped me with some of the basic tools that I need, columns and stuff like that, because they have been developing a new technology, wanted me to use the technology to see if this works for this model. My funding comes from AUP, and then I have multiple internal research grants that supported this research. So in this talk, I would give you a general overview of the problem that we are facing. And then I will talk about existing methodologies, and then give you an introduction to specific technology that we're going to discuss, which is hydrophilic interaction liquid chromatography, and then couple it with the detector that we are using. This is called a CAD detector. And then give you an overview of the analytical strategy that we employed for developing the method. And then we'll discuss some results that we got from this research and then give you some additional thoughts. So to start with, I will talk a little bit about nectars. Nectars are clear as water. You pick any flower, you would see that there is a nectar which looks very similar to water. It's very light, its viscosity is low, and you would be able to collect it pretty easily. Chief constituents, in fact, about 90% of this material is mass over mass, just sugar, glucose, fructose, and sucrose. And nectars are important because they help in pollination. Most of the pollination happens by bees, and they need food. And so they collect the nectar, bring them back to the hives, and then convert it to honey. And that's how they store the sugar. But if you look at the type of flower we are working with, all of these flowers, or most, I should say, most of these flowers have colored nectar. Now, the natural habitat is all throughout South America, but we get most of our material from northern Peru, where Tom travels and then he collects the material and then bring them back. Because I'm showing this area doesn't mean that the species is only connected to this. It's all over the South American countries. If you want to know more about it, you can go through the link in our website. He has distribution maps of different species throughout the northern and southern Americas. For about 20 years, people have been doing research in this particular area. And colored nectar is very, very uncommon. There are only about 80 species in the world where people get colored nectar. So not a lot of people work on them. Tom certainly does, and you can see he worked on distribution and ecology and evolution of colored nectar. He also looked at a specific colored nectar called Mauritian colored nectar. And unlike regular pollinator like bees, you can see in this particular colored nectar, a gecko is going inside the flower, which tells us that some flowers and plants find a different type of pollinator that can go in. This is certainly going to be interesting as we go forward, because the sugar distribution is somewhat different in the colored nectar versus the clear nectar. And I'll show you how we get to that conclusion. But for now, just know that the colored nectar is pigmented and it's very uncommon. So it's important to know what is in that nectar that is different than other nectars that we see. I took some of the pictures from Tom's website. This, as you can see, when the nectar starts, it's going to be clear. But over time, as you see, day 1 to day 2 to day 3 flower, we see a change in the nectar, and the pigment kind of takes over and makes it darker. I have seen a range of light orange or even yellow to dark red. We know that this nectar is also pH sensitive. The color changes between yellow to dark red when we go from acidic to basic. So using this preliminary information, we wanted to develop something that is robust and quantitative so that we can determine the sugars from the nectar. And we hoped that when we establish this method would be widely applicable to other model systems. So if we wanted to go to leaves, or if we wanted to work with other models, we could easily transition into those models. Now, sugars are typically determined from various other disciplines of science, especially important in food chemistry, but used in environmental science, certainly in medicine, from physiological system, from general food and agriculture, as well as, this is somewhat unusual, but archaeological studies sometimes work with sugars. So it's quite important to know what is there in the complex matrix and how much of it is present. If we look at the existing literature, and I don't mean to be exclusive, this only covers a few of the techniques that are available. There are many more, but they generally canter around 3 different areas. We have spectrophotometric methods. These are age-old, about 70, 80 years is gone that when we use spectrophotometric methods. Even today's research, people try to develop spectrophotometric techniques to analyze carbohydrate. And then there is this big area in last 20, 25 years that became popular using LC-MS to run carbohydrate analysis. Between the two, we have another technique called high performance anion exchange chromatography that connects with a pulsed amperometric detector. And this proved to be quite successful in looking at sugar. Now, the point that I want to make over here is almost all of these methods have limitations and problems and issues. None of the methods are universal. And as an analyst, we have to be careful and see what we can live with and what we cannot. Could they give you reasonable result? Yes. Are they easy to apply? No, especially LC-MS. This is not for faint-hearted people who try to use them for carbohydrate analysis. There are a lot of complications if you try to LC-MS using for carbohydrate analysis. The basic strategy is very simple. We have to think about the extraction strategy from the complex matrix. Then we will separate because we're interested in individual sugar molecule, and then we will detect them, right? But there are problems in extraction. There are certainly huge issues with separating these sugars, especially when they're present in complex matrices. And then signal intensity could be a really big challenge, depending on what type of detector you're using. Now, because I'm guessing that most of you are undergraduate students, perhaps you are not quite aware or have seen high performance liquid chromatography. So I put together a very quick figure, and then I would explain how high performance chromatography works. Now, if you're familiar with this technique and you know, please bear with me for a few minutes. I promise you I'm not going to be long. In general, high-performance chromatographic techniques, especially liquid chromatography, we have different types of solvents. These solvents are pumped and they go into a proportioning valve where you mix this solvent. So you change the polarity. You can do isocratic separation where the mixing is constant over time, or you can do a gradient, which is changing these mixing ratios over time, right? Then they go to the input check valve, and then through the output check valve, goes through a pulse damper. Filters are relatively uncommon in the new instrumentation. Pressure transducers are, you would not be able to see it, goes to the injector, and then it goes to a separation device. For liquid chromatography, the separation device is typically known as column. Gas chromatography too. The types of columns are very different in liquid chromatography versus gas chromatography. Gas chromatography columns are very long. If you're using a capillary system, it could be 30 meters long. Whereas liquid chromatography columns in current day and age only going to be about 10 centimeter or a maximum of 15 centimeter. People are trying to get away with a 3 centimeter column, very small column. Okay, so that is the basics. What we should be focusing on is how do we do the separation in here, and what kind of solvents can we use to kind of manipulate the polarity of the analytes that are injected and then separate them in the column. So essentially, we'll talk about the stationary phase and the mobile phase. The instrument that we used is called the Ultimate 3000 instrumentation. You can see this is modular. I have 4 different solvents. The 5th one is just a rear seal wash. There is pump inside, and the pump is pumping, and then here is the solvent sample tray. The injection happens here. The column is stored inside, so the solvent, mixed solvent, comes in, goes over here to the column. You can't see the column, I'm sorry. And then it goes to the detector. Now that is a different detector. It's a photodiode array. The CAD detector looks like this. And I'll give you a brief overview of how the system works within the CAD detector in a few minutes. So let's first look into the stationary phase. How do we separate? What do we have to have so that we can keep the sugars inside the stationary phase for some time? There has been research, especially in last 10 years, we have learned a little bit about how to work with hydrophilic interaction liquid chromatography. This was coined somewhere in 2004, 2005, but then new developments came in. Multiple different people published in these areas. They were trying to all figure out what type of methodologies are most reliable to detect sugar using this type of stationary phase. So this particular phase is somewhat unique in the sense that it uses normal phase adsorbent. Now, what is normal phase? Normal phase is mostly bare silica particles, okay, something that is highly polar, okay. It uses the eluent. Eluent is the solvent system from reverse phase liquid chromatography. Now, why is this important? So that you can ionize rather easily. And then the analytes are often charged and polar molecule. So bring in the concept of ion chromatography. Helic kind of overlaps with 3 of them. So there is a sweet spot if you could be able to choose your analyte carefully if you can find a reasonably stable stationary phase and an elution system, you should be able to separate the sugar molecules without any problem. If we look at the chemistry of the stationary phase, these are what is inside the column, right? So we have a steel container essentially that is packed with particle, right? What type of chemistry should be there in the particle? So for bare helic column, we have two functionalization, silanol and siloxane. Okay, these are highly polar molecule, and I'll show you in a few minutes how these polar molecules allows a layer to be formed. Okay. Then people started modifying this type of moieties, and they ended up with an amino module. And then more recently, we saw that there is a zwitterionic phase with amine and sulfonic acid that can be used for stationary phase. Okay, so helic, zinc helic or helic Z, and amino. So how does the separation really happen? Well, inside the column, as I mentioned a few minutes ago, we have packed particles, right? So those packed particles have bare silica, and that is going to be the helic phase or functionalized silica, which is also part of the broader umbrella of helic phase. Whenever we try to separate, we have an aqueous layer formed on top of the helic phase. Okay, the molecules being polar are going to be carried using an organic and aqueous phase. The analyte will distribute themselves or partition themselves between the water and the organic layer. And then when the elution comes in, when they partition into the water layer, they're interacting with the helic phase. Then they will come back to the mobile phase and get out. The separation is, or partitioning, is strictly governed by electrostatic interactions as well as hydrogen bonding. So what's the catch? I mean, it looks fairly simple. We have a solid column. As long as we can build the water layer, we could be able to introduce the analyte. Well, the problem starts at the beginning. Where do we typically dissolve sugar? In aqueous system. Now, if you have too much water, this is not going to be helpful because your organic mobile phase now becomes a smaller percentage, and the analyte would not partition into the water layer, which means the analyte comes in, it zooms through, and it goes to the detector. So there is no separation at the end. So we need to figure out a way to dissolve those sugars in a, let's say, an unfavorable solvent. But if we try to do that, then we start seeing precipitation, syrup in liquids, all sorts of problems. So what we did is, through our work, we figured out a sweet spot that might work okay for loading the sample with a reasonably small quantity of water. There is another problem. Helic phase is highly polar. If you try to add a substantially polar mobile phase, you are going to start washing out the particle themselves, which means the integrity of the stationary phase is going to get compromised. That's not advisable. So we need to keep the aqueous layer stable, but we can't use too much of water into this system. So let's see how we should proceed. Here you see that I have two different types of particles that are typically used in this type of columns. This is, by the way, the columns that we use for separation. When any separation happens, use reverse phase or normal phase, we try to load a band of the solvent. So essentially a molecular band is coming through and getting loaded at the beginning of the column. There is no way that this band can be stopped from broadening. It is going to broaden as it goes through. As you can see, bandwidth kind of increases over time, right? So at the end of the day, the molecules are all loaded initially here. They start interacting with these particles and they will separate from one another. And then each band will come at the end of the column, right? They will take different path and eventually they will give us a response. Now, if you look at the response, you see that these responses are quite broad. Without going into too much details, I can tell you that broad peaks are often problematic for quantitation. Sharp, narrow peaks are always useful if you're trying to develop a quantitative method. So initially, we started working with fully porous particles. What is a fully porous particle? Well, within this particle, solvent can go in and out. Analyte can strongly interact with this type of particles, and then they will be released. In recent years, a new type of particles were developed. These are known as solid core particles or core shell particles that gets packed within the column. The advantage is enormous. In solid core particle, when analyte starts interacting, this band broadening is significantly reduced because the analyte is not fully partitioning within the stationary phase. Instead, they're interacting very strongly with the outermost shell. The core remains solid, and the solvents, which kind of creates an enormous back pressure. And when we talk about back pressure, we are talking about, let's say, 10,000 psi or 13,000 psi back pressure. That pressure issue can be addressed using the solid core or core shell particles. So I started working with this about 4 or 5 years ago, and I found them to be a fascinating model to do the separations. As you can see, multiple paths are very common when we have porous particles. Not that they are not useful. There are still 1.7 micron particle that are being heavily used by academic and industry to do some really nice separation. And the smaller you go, the better separation you can achieve. And you can't do a really small particle with core shell. Core shell works well with about 2.7 micron particle size. This can go even smaller, and I'll show you why smaller particle sizes are important in this type of separation in a few minutes. Okay, so bottom line is core shell particle can reduce the back pressure issue, which makes them valuable. It also keeps the peaks quite narrow. So if you're planning on quantitating something, this could be quite useful. Now, people who work in analytical chemistry, they always talk about this particular equation. Now, I don't want to go into the details of math associated with it, but what I just kind of brush is we, as an analytical chemist, we always look at theoretical plate height, and we have a gold standard to reduce the plate height. We know that smaller plate height and higher plate count is better for separation. Now, some people in your generation, when you do analytical chemistry, you would perhaps come back and tell me plate height is not going to be a useful parameter. And we completely understand that. But industry hasn't even moved from looking at the plate height yet. That's why we use this particular obsolete term. Now, what it means is it depends on multiple parameters of separation. The first one is multiple path effect, which I showed you a few minutes ago. Analytes can travel using different paths, and if they are traveling between different paths, naturally they will broaden. And if they become a broad peak, then separation will have issues. The B term is associated with longitudinal diffusion, and then C's are nothing but the mass transfer between the stationary and the mobile phase. Because the way this separation works is we have our stationary phase, which is fixed in place, and then mobile phase is carrying our analyte. Those analytes are constantly interacting with our stationary phase and then slowly moving forward. So in the process, they start narrow and then they become broader. So what are we trying to achieve? A good separation with a reasonable particle size. We know that for particle sizes, as you can see, if I look at the linear velocity versus h, as we increase, as we increase particle size, we are going to see much broader peak because h is going to be higher. Okay, so smaller particle size, great. Larger particle size could be a problem. The take-home message is, how do we deal with the back pressure issue? And core shell particles kind of address that particular problem. We know that core shell particles are going to be really useful for this type of separation. Smaller particle size is good, but I will show you in my research that I really worked with a very small particle size, and that did not solve the problem that I wanted to solve. It still has a huge back pressure issue that I could not be able to resolve when I stayed with the smaller particle size. Besides, the chemistry was different with smaller particle size. Now let me give you a brief overview of the detector that we need to use. So first part is mobile phase solvent, which we did not discuss yet. I will show you when I show you the data. We discussed the stationary phase, which is going to help us separate the compound. And now, how do we detect it? There are multiple different types of detectors which can be used with this type of chromatography. The most common one is refractive index detector. This detector is very general. However, if there is any change in the mobile phase composition, we are going to see a huge drift in the baseline. And if that happens, again, your separation is going to suffer. The second batch of detector is evaporative light scattering detector. In this type of detector, very commonly used by academic and industry. This detector is generally quite useful, but it looks at a very narrow group of molecules, and I'll show you what I meant by narrow group of molecules in the next figure. And then the type of detector we have been using, this is the charged aerosol detection. Now, I'm not touching base with liquid chromatography mass spectrometry. That is always one of the main choice for scientists these days because of the extreme sensitivity. But with extreme sensitivity, you run with other issues such as baseline drift, matrix effect, ionization problems. And not that I don't like mass spectrometry. That is part of my research. I do use mass spectrometry all the time. But it is important to understand the caution associated with quantitation involved in mass spectrometry. It's not a black box. It has several different problems that needs to be addressed beforehand so that we can develop a reasonable quantitative method using that instrument. All right, so how does this CAD detector work? Now, at the end of the column, we are getting liquid that contains our analytes, right? We separated the analyte inside the column, so they are now coming as a function of time. Molecules that are traveling faster, they will elute faster and they will come to the detector first. When they come, it comes through a tube and it goes to nebulizer and impactor. This is connected with a gas line, nitrogen. Dry nitrogen is used in this process. You can see nitrogen comes in and then it divides itself into two different channels. The first channel goes forward and impacts with the eluent that carries our analyte. Okay. Now, when it impacts, it nebulizes, right? So nebulizes is forming aerosol. And there are three different groups of aerosol that can be formed. Some aerosol could have a very small size. Some could have a very large size. Not all of them are going to survive in this process. So you can inherently see that there is a problem, right? We are getting our analyte and it is being nebulized, but not all of the analyte that is being nebulized are going to be detected. The larger sizes are going to form droplets and get out. Then we have very small size aerosols that will also not survive this impact and they will also drain to the west. Only a few analyte is going to travel. The analytes, they are in the middle region, right size. They are going to travel to the next phase. As you can see, these are different colors, so different times, different types of analytes. And then they will reach the mixing chamber. Okay. Let's take a pause here and look at the other side of the nitrogen and what's happening with the nitrogen that gets diverted through the second line. So nitrogen is coming in and then it encounters a corona discharge needle. The job of the needle is to just transfer charge to nitrogen molecules. Okay, so charged nitrogen molecule now enters the mixing chamber from the other side. Okay, so analytes got nebulized. They are in the gas phase. Now they are kind of dry and they are entering the mixing chamber. From the other side, we have nitrogen coming in. Those are charged. Now, organic molecules tend to retain charge way better than nitrogen. So what's going to happen is this charge that we see that are coming through now get transferred onto our analytes. More analytes, more charge. Less analyte, less charge. They are then going to travel through an ion trap. These are charged particle or charged analyte at this stage enters the ion trap and becomes stabilized here. And then the gas pushes them to an electrometer where the analytes gets discharged. So if I have a lot more analyte, I am going to get a lot more discharge. If I have a small quantity of analyte, I'm going to get small discharge in here. And then these charges in the electrometer is essentially give me a signal. More molecules, higher signal, less molecules, lower signal. What would be the catch? Well, what happens if we get the molecules inside the mixing chamber and it doesn't prefer a charge. Would we be able to detect it? No. But fortunately for us, most organic polar metabolites and sugars that we're going to be working on tend to retain these charges. So the detector can be useful in detecting those type of polar metabolites. What are the general challenges that we deal with when we develop this type of methods? Dissolution, which we discussed before, we can't have a lot of aqueous material in the system. If we do have a lot of aqueous material, it is not going to retain in the stationary phase. That's because we are starting with a very high concentration of the strong solvent, which will kind of stop the interaction between the analyte and the stationary phase. In the chromatography side, we have to think about stability of the helix phase and the type of additives that we can add. Too much water, the helix phase is going to get destroyed because it's going to take the particles away. Additives, we need to be very careful here because CAD likes volatile additives. So something like ammonium acetate, ammonium hydroxide could work. But if we started adding non-volatile component, they are going to go ahead and create precipitates and solid particulate matter at the tip of the needle that allows the nebulization to happen. Huge problem to deal with. Okay. And then finally, we need to think about charge formation. If most of my molecules are larger, becoming larger when they are getting nebulized or remaining smaller, we have a serious issue. We can't really deal with quantitation if we have different type of distribution. So we want most of our molecules to be in that middle range so that they can get detected. So what type of column should we use? Well, we have a variety of different columns that we started working with. We have a Helix AccuCore. That is what we started with. Then we went to a Helix Kinetics. We used an amino functionality that is called a Luna column. And then we ended up using the Helix Z column, which is a porous shell, which is a core shell type of particle. So we have mixture of carbohydrate. We analyze them in individual columns and then evaluate the analytical figures of merit, figured out the pros and cons, and then get to the method that we could be able to utilize. And then eventually they all went to this detector. In most of our work, you are going to see glucose, sucrose, mannose, and fructose. Sometimes you will see that I have used lactose, and in other cases, you'd see lactose was not used. The work was carried out in different phases, and I will show you the first, second, and third phase of developing this particular method. Okay, let's start with something that was simple. We started off with a mixture of mannose and sucrose at the beginning. We used a AccuCore column. This is 5 centimeter and 2.6 micron core shell particle. It's 2.1 millimeter diameter. We kept the column temperature at around 40 degrees Celsius, and we used 94% acetonitrile, which is our weak solvent, and 6% ammonium acetate buffer as our solvent B. This is an isocratic separation, and the response shows that the mannose and sucrose are being separated. You can, the nice thing about this is you can see how quick the separation was. Between 1 minute, we were done. The void time, which means the non-retent species, came out within about 0.2 minutes. We tried different flow rate, and I think this one is carried out in about 1 milliliter per minute. So we're quite happy with our first level of separation. The different lines show different types of concentration that got injected. When we inject smaller amount, we get a smaller peak. When we inject larger amount, we get a larger peak. Using this, we can create our regression and essentially calibration. Then we started fooling around with some other sugars. Here you can see we have fructose, mannose, and glucose, and we changed the pH a little bit, tried to work with 90% acetonitrile and 10% water. So we increased the strong solvent a little bit, increased the flow rate also 1.2 milliliter per minute. What did we see? Immediately we saw broader peaks. Now we know that glucose and mannose are quite similar to each other. And we knew that it would be very hard to separate glucose and mannose using this type of stationary phase. And there was no surprise. They were on top of each other. What was surprising is when we look at monosaccharides and disaccharides, we saw that using the initial method, we can't really separate actually, we cannot really separate fructose from sucrose. And that was alarming. We knew that we are having some issues with this particular particle size. So naturally, we thought, okay, if we go to a smaller particle size, the resolution of the peak is going to be better and we could be able to see them. So we started using a slightly different column. This one is with 1.7 micron particle size, very small and porous, right? Same thing, 90% acetonitrile, 10% water. Everything else remains same. We did individual injection, did not see much separation of the two sugars that were giving us problems. Okay. So at this time, we're kind of at a loss. We thought, okay, well, if we change the parameters, we should be able to see at least something. But it looks like it is not changing at all. Now, why could that be? It could be because of two reasons. Number one, maybe 10% water is too high for this type of this type of analyte, and having 10% water in the mobile phase may be too fast for the analytes to move. So we started, working with some other phases. But before we go into the phase, we took a step back and started thinking about what could be changed in each individual stages. So we started figuring out 4 different parameters. We saw there is a phase issue, there are pH and additive issues, dissolution problem, and there could be some issues with flow rate. We knew that the phase, if that is not conducive, we are going to have poor separation. If the pH and the additives are not appropriate, we will end up with separation issues as well as detection, because detector might not like what we are adding, and the ionization would not happen if the correct additive or pH is not maintained. Dissolution will certainly be a problem because it's going to give us a splitted and broad peak because it's not getting loaded properly in the front of the column. And then flow rate is certainly an issue that is associated with mass transfer. And if the flow rate is not appropriate, we're going to see a broad peak. In some cases, you did see a broad peak so far, okay, but we're already towards the tail end of the flow rate. So we can't modify that anymore. We need to work with other parameters. So the next possible thing that we could do is change the functionality. And we started working with a different column, a mean functionality. This is run in 75% acetonitrile and 25% water in an isocratic mode. And we loaded mixture of glucose, fructose, and sucrose. And we immediately started seeing broad peaks. Now remember, we are already at the tail end of the flow rate. We can't really manage the flow rate anymore. So it was kind of clear to us that it is going to be harder and harder to separate, these groups of analyte if we stick to this type of functionality. However, we changed a little bit in the mobile phase and tried to see if this separation improves. And no, it did not. The peaks become more and more drastic. The baseline started drifting and we have very broad peak. This is not a nice separation. If we are trying to work with the complex matrix, and if this is the signal we get, we're doomed. We can't really have reasonable quantitation using this type of peaks. We need to do better. Now you can see that here we actually changed the flow rate. We have a variety of different flow rate to try and see if this improved. As soon as we increase the flow rate, these all jumbled together and you would see no separation at all. So we found out that additives and things like ammonium acetate for amines are not really useful because it's not really conducive to this type of systems. We moved on to a third phase, and this is the Zik helic phase, which is deuterionic, right? So we have an amine functionality and we have a sulfonate functionality. When we try to look for separation, this is again a 2.6 micron helix Z column. So again, coarse shell particle, simple format, use acetonitrile and 20 millimolar ammonium acetate. We found the peaks to be sharper. In fact, quite nice compared to what we have been getting. We found fructose, we found sucrose individually and from the mixture. You can see the time has increased a little bit. We are seeing things about 5 minutes. So we knew that the only challenge is the glucose peak. Right now, why is glucose showing this type of splitted peak? Well, the explanation is very simple. When you study carbohydrates, you will see that glucose can form alpha and beta anomers. And the exchange of alpha and beta is quite rapid. In this stationary phase, we are seeing both of those anomers, which are interacting slightly differently with the stationary phase. So the band essentially becomes slightly broad, but not broad enough to give you a single peak. Instead, it gives you a dip. Once we figured that out, our goal was to ensure that we wanted to have only one specific anomer in the medium. And so we started fooling around with the pH parameter. But we have to be careful because CAD is also playing a role. If we try to modify the pH too much and bring in quite a few foreign additives, we are going to see there is a drift in the baseline. So once we started increasing the pH, we saw the peak shapes are improving. The analysis times kind of change a little bit, but peak shapes certainly improve. We also noticed that when we change the initial solvent system to 65 and 75% acetonitrile, this data only shows 75%, but later on we modified this to have about 65% acetonitrile, 35% water. We found this to be quite good in terms of peak shapes. So high pH and changing the initial solvent system helped us get to where we want to go to. So here is the nicest separation using acetonitrile and high concentration of base in water. This is also isocratic. It's a little long, as you can see. Sugars in this case was dissolved in 75%, but later on we found that we may have to vary this to stay between 65 and 70%. The most interesting thing is the split peak issue that we were seeing for glucose. That is now gone. pH resolved that particular issue. So we started looking at the calibration curves, and we got reasonably good calibration curve for each of our sugars. As you can see, fructose, glucose, and sucrose all gave R square equals 0.99. Very good. Now, when we started working with the nectar, we did see slight variation, but still quantitation could be done within this. This is not really extremely nice peak, but considering where glucose was, this is still useful. There is one thing that I want to point out here. You see, the two look slightly different. One is phase separated and another is not. Phase separation occurs when we use a very high concentration of acetonitrile. And we suspect that if you try to use, or if we try to use high concentration of acetonitrile, some of the components are no longer staying in the acetonitrile phase. Even if acetonitrile and water is soluble, we are forcing those molecules to go and kind of get suspended into the aqueous layer, which is a problem because homogeneity is the first step in these processes. If the sample is not homogeneous, we're going to run into problems. These are some of the flowers that we worked with, 4 different species. You can see Calliantha, Haltomata calliantha, Haltomata quipuskoi, Haltomata wigendiana, and Haltomata grandiflora. These 3 produced colored nectar. Grandiflora produces a clear nectar. We started looking closely at the calibration curve. And we saw that even if we are getting a good regression, it is not perfect. So we started investigating as to what is going on with this trend. It kind of looks like it's a dip and then there is an increase. It's a dip and then there is an increase. It's linear enough to be used, but it's not perfect. So we went back to the detector and started looking at, is there something that we missed? And we saw that, yeah, there is something that we should have to consider. And that is, this detector is not really a linear detector. It's a non-linear detector. Non-linear detector, it's a mass-sensitive non-linear detector, which means the regression may not be quite simple. And we found out that a linear fit is not the right way to do this type of calibration. The better way to fit this type of calibration is through quadratic formula. So once we realized that, we started looking at individual sugar molecules, and we found that a quadratic fit is much better in fitting the data. The fit is higher than R square 0.99, and 95% confidence interval shows that this data set is quite reliable. So that's our regression curve. It's not really linear, but it's quadratic, almost all of them. And the dark points are essentially check standards, which runs between the standards and the samples to ensure that the integrity of the calibration is held over time. Okay, and it shows you they fall right on the calibration curve, exactly where we want those concentrations to be. When we look at the percent RSD value, which is a measure of reproducibility, we see that this is very, very tight. Often it's within 0.1 to 2%. All right. So at the end of the day, what did we get from the Altomata species? We analyzed 4 different types of plants. They were in 2 different phases. One phase is called hermaphroditic phase, and the other is called pistillate phase. Pistillate phase is the beginning of the flower phase, where there is no pollen going into the nectar. So the nectars are pristine. In hermaphroditic phase, pollens from the anther can go into the nectar. So you might have some contamination. Now, Dr. Mayon collected both hermaphroditic and pistillate phase so that we can compare and see what the variations are. And we were able to detect fructose and glucose in almost all of the nectar, right? So lower limit of detection is good enough to detect them. The trend is kind of different. In the clear nectar, which comes from grandiflora, we see that the amount of sucrose is high. Whereas grandiflora is not detected in Caliantha, grandiflora is, sucrose is really low or not detected in Caliantha and low in Wigindiana and Koipuskoi. So we started thinking, why? Why do we see a different pattern in sucrose concentration? And why are glucose and fructose kind of varying between the clear nectar and colored nectar? And we come up with the explanation that the pollinators play an important role in this. If we look at pollination using bees, which happens in case of grandiflora, we see a higher amount of sucrose present because honeybees collect sucrose, glucose, and fructose all. Whereas if geckos are coming, they would only work on monosaccharides. So they need fructose and glucose. So the plant is smart enough to go through an evolutionary process so that they can manipulate their sugar metabolism in nectar. Now, more details needs to be added to it, and we are still investigating, so I can't give you more on this at this time. We work with a significantly large number of grandiflora sample recently to try and take a look at the pattern, and we saw our previous pattern holding. Fructose and glucose to be in smaller quantity and sucrose to be in high quantity, n equals 7 in this case. And the error bars are essentially showing the standard error to mean. This is something we are putting together in a publication. So what inferences can we draw from all this? Number one, Helix Z is a suitable technique. It is able to detect monosaccharides from complex matrices. However, we need to carefully consider the methods. CAD demands additional attention in terms of mobile phase solvent choice. We need to think about additives that can be added or cannot be added. And it is especially important when we are looking at carbohydrate mixtures. Helix separation depends on uniformity of the aqueous layer. It is absolutely of paramount importance that we pay special attention to this particular part. If the layer is non-uniform, we are not going to get a good separation. Now, how do you achieve that? Over time. You need to give enough equilibration time so that that layer can be formed and become stable. We still need to do additional column stability studies because we have used a relatively high quantity of base. pH was high. We needed to figure out how the silica particles are going to hold up. Now, there are some studies published by the vendors which shows that this runs for about 14,000 cycles within their facility. We haven't run that level of study yet. We are getting good enough amount of injections, but we need to do a little bit more to find out. At the end of the day, we know that Helix Z can provide a robust quantitation from low amount of sugar from complex matrices. Could it be plant-based? Could it be mammalian system-based? Either way, it is going to be able to provide you with a reasonable data if you're careful with the method development part. With that, I would like to end it here, and I hope you enjoyed. I know it's evening time over there and you can spend your time doing several other things, but you decided to be here. So I hope you enjoyed and learned something from it. That was my research group pre-COVID. Many of them are in different places now. My recent research group is a bit smaller compared to this. After COVID, we have a bit of drop in number of students. I work with one student at this time. All right, with that, I would like to thank you. And if you have any questions, I'll be more than happy to answer them now. Thank you for your patience.
Well, thank you, Professor Chakraborty, for such a resourceful and engrossing lecture. I strongly feel that our student must have learned about some aspects of HPLC and their application in separation of mixture of carbohydrates and how to apply these aspects in science for real life. That is very, very important. And I think the students may have one or two questions. So please.
Absolutely.
Students.
I'm ready.
Yeah, students.
Yes, sir.
Yes, please interact with Professor Chakraborty.
Yes, sir.
Don't feel shy. I
to
speak
interact with
Bengali.
him.
If you want to ask a question in Bengali, I'll be more than happy to answer it.
Yes, sir. The first question is, how are the core shell particles synthesized?
Those are mostly in the engineering facilities. Mechanical engineering typically plays roles in synthesizing large-scale particles. Unfortunately, these are trade secrets for most of the company. If you go to Agilent or Thermo, they will tell you what is in there, but they would never tell you how to synthesize these particles. But you can take bare silica particles from a vendor, and then you can functionalize them. The biggest challenge is maintaining the uniformity in the particle. Let me go back to a slide to show you something that I may not have pointed out. Here. So if you look at the top one, the core shell particles are mostly uniform, right? But if you look at the fully porous particles, there are different sizes. So there is a size distribution in the porous particles. And if you are doing it on your own in a small scale laboratory, the chances of getting the particles to the uniform size is a bit lower because it's essentially done in a essentially chemical chamber. I'm not sure whether that answered your question, but that's what it is. We don't know much about it because it is mostly trade secret for the vendors. But these are very similar to the silica that you use if you use open tubular column chromatography. Does that answer your question, at least partially?
Yes, sir. Yes, sir.
Okay.
Yes. Sir, the next question we have is, how does the electrometer detect particles?
They don't. What they do is they detect charge. So what happens is these have diodes, and what they are trying to do is they're looking at the charge. Now remember, each of our analyte molecule is getting charges from nitrogen. right? So if you, let's say if you have 10 analyte molecule, they will accept 10 charges. If you have 30 analyte molecule, they will accept 30 charges. And then when they are going to the electrometer, those 10 analyte molecule will discharge their charge. And the charge is what the electrometer is going to see. So it is not seeing the analyte molecule, but seeing the charge that is present on the analyte molecule.
Thank you, sir. The next question we have from our audience is, why is the column temperature kept at 40 degrees Celsius and the flow rate maintained at 1.2 milliliters per minute?
So when I showed you only one set of column temperature, and that is 40, but we did some tests between 35 and 45. I can tell you why we did not use room temperature, because room temperature kind of varies in our laboratory. So we wanted to have it separated at a slightly elevated temperature. The point to use 40 degrees is to stay slightly above ambient temperature, and 40 degrees is relatively standard for any separations that we do. Doesn't mean that we can't go for 45. We have gone to 45 and we did not see much variation in the in our separation efficiency. 1.2 milliliter is the sweet spot where we see reasonable separation of analyte. We have used 0.3 milliliter, 0.6 milliliter, 0.9, and 1.2. And remember, I showed you a figure, or maybe the equation. Hang on just a second. Here. So CS and CM are mass transfer coefficients, right? So what they are trying to tell us is the analyte, when it moves through the stationary phase, is coming with a mobile phase, and it is interacting with the stationary phase and then going back to the mobile phase. If we keep it too slow, the band will invariably broaden. If we try to do too fast, we are going to see a huge amount of back pressure, because you have a very narrow bore column and we are trying to force too much solvent at a high flow rate. So back pressure is going to increase in such an alarming rate that we might end up with a shutdown of the instrumentation. 1.2 is the sweet spot which we found work for this separation. It's associated with the mass transfer coefficients.
Sir, next question we have is, are there any limitations of the Van Demeter equation?
Several different types of limitations, but not specifically with the equation itself, but with the methods that we develop. Van Demeter equation just tells us what we should look at, right? So we should look at eddy diffusion coefficient, which means, is it going to take a lot of different types of multiple path? What Van Demeter equation gives us is a theory behind band broadening. So if we want to have a narrow band, what we should look at is tighter particles. We should look at longitudinal diffusion of the analyte, which is relatively different between the reverse phase separation versus the separation we are doing. If you're doing reverse phase separation, analyte directly interacts with the stationary phase. But in our separation, analyte interacts with the water layer and not directly with the silica when it gets retained in the column. And certainly mass transfers. If the mass transfers are not happening because of the mobile phase velocity, we are going to see almost no separation. So method limitations are certainly a factor that needs to be considered. This just gives you a broad overview of which parameters are critical for the separation itself.
Sir, next question we have from our audience is, why is the sucrose more in J. grandiflora?
Right. So I think I briefly described it, but let's go back to the slide. So this is a clear nectar, right? The pollinators are bees. If you remember what my Nectar 101 slide looks like, they were all clear, right? Most of the time, these flowers are pollinated by bees. Now, when bees collect nectar and convert it to honey, they wanted a mixture where they have a lot of sucrose. However, when you look at the colored nectar, in every colored nectar, you would see that glucose and fructose are going to be relatively higher compared to sucrose. Now, pistillate phase is the early phase of the development. So sometimes you may not have enough sugar to work with. But when they go to the hermaphroditic phase, that's when pollens are dropping in the nectar and bees are coming to collect, or any other pollinators are coming to collect the nectar. So we believe that the amount of sucrose is higher because those flowers are pollinated by bees, whereas all colored species will have other pollinators like geckos, and they don't care too much about a disaccharide because they will have to enzymatically digest the disaccharide to get to the monosaccharide. And then eventually that monosaccharide is going to be used in metabolism, right? Whereas in case of bees, they want the disaccharide so that they can convert it to honey. That's why and this is a hypothesis, that's why we believe that the amount of sucrose is higher in grandiflora.
So what are the future prospects of this research?
We know very little about any species that has colored nectar. As I pointed out, there are about 80 species in the world that produces colored nectar. So one strong aspect is to know why the chemical profile is what it is. In general nectar, you will find different types of sugars. And in this particular species, or this particular genus contains a variety of different species where we don't know what the chemical makeup is. So it's just natural inquisitiveness for us to know what it would be. Now, there was a paper recently came out from University of Minnesota who started looking at why this particular pigment is going to be formed. You can imagine that if you have a transparent nectar, you are looking at systems that would be fairly simple in terms of biological, biochemical reaction or networking pathway. Whereas if you produce something that is colored, you are involving complex molecule. Why would a plant try to produce a complex molecule in the first place? It requires significant amount of energy. So is there an evolutionary trait in this particular plant that forces them to spend their energy in producing the compounds that they are producing? The group from Minnesota claimed that this particular compound is called nesocodone, and they showed a strategy to detect this type of compounds. We are still working on it from the natural products chemistry side to try and figure out what the compounds are in our nectar. They worked on Mauritian species and another species of Altomata that's called Herrera. So it's naturally interesting to us to know more about these type of unique systems. That's my general interest. It has nothing to do with medicinal value. It has nothing to do with anything else. Although I must say that this particular species produces a fruit, and that fruit is consumed by people in Peru and some of the ethnic food, even in the United States. And the nutritional values are not known. So we have been working on that aspect too, to try and figure out if this particular fruit is different than any of the other fruits in the Solanaceae family, where this belongs. Nightshade.
Thank you, sir, for patiently answering all our questions.
My pleasure.
Now
Anything else?
No, sir. No, sir.
Okay, if more questions come in, feel free to email it to me. You can find me in the CCSU website, or you can go through Dr. Ray or Dr. Ganguly. They can forward me your questions if you're interested.
Absolutely, sir. Now
Okay.
I would like to call upon our esteemed professor, Dr. Shonjeev Ganguly, to deliver the vote of thanks.
Okay. Well, so this is the official vote of thanks. I would like to express, first of all, my heartfelt gratitude to Professor Sourav Chakraborty for taking time out of his busy schedule and delivering such an enriching lecture for our participants. I'm sure they have learned something from it, and I hope they are inspired by your lecture. It's a new thing for all of them. In fact, HPLC, probably they have not studied right now. So it's a new thing. They must have learned something. Well, I want to thank Father Principal, Dr. Dominic Savio, for his constant support. Vice Principal, for his support. Of course, the head of the department, who was involved in organizing the entire program, and the faculty members of the department for their support. And lastly, and most importantly, I would like to thank our students, the organizing committee, and the entire Department of Chemistry for the hard work, planning, and dedication in making this seminar a success. Thank you.
Thank you, everyone.
Thank you.
Thank you, sir. With this, we come to the end of today's webinar. Thank you everyone for joining with us as we start our journey of RACB 2022. Stay tuned with us for the upcoming webinars. All the registered participants are requested to fill up the feedback forms from the link given in the live chat to obtain their e-certificates. All of you are also requested to subscribe to our YouTube channel to get the updates of the upcoming webinars. Thank you once again.
Bye-bye.
Okay, thank you. I'm leaving. Thank you, students.
Thank you, sir.
Thank you,
Thank
sir.
you, sir.
Thank you, sir.
Thank you, sir. Thank you, sir.
Sir, you are muted.
Sir, you are muted, sir.
What I was saying, it was a great show. Thanks to the organizing committee, everyone, RS sir and HD sir, all our departmental faculties, especially the organizing committee students. And it was a great show. Be ready for tomorrow. Okay.
Thank you, sir.
Yes, sir.
Thank you, sir.
Yes, sir. Thank you, sir.
Thank you, sir.
Yes, sir. Good night, everybody.
Good night, sir.
Good night.
Good night.
Good night, sir.
Good night.
Good night,
Good
sir.
night.
I'm stopping the stream as well as this meeting.
Sure, sir.
Okay, sir. Sure,
Yes.
sir.
Yes, sir. Okay, sir.